Welcome to a different kind of garden. I'd like to introduce our speakers for tonight. Elizabeth Woodcock. Elizabeth is an ecological gardener, a master composter with Garden Organic, and a writer with Frontiers magazine. To her right is Anna Canning. She is a herbalist, forager, and former translator of 4 languages, I believe. Together they will share the wonders of microbiomes and their exploration of this new science. So please put your hands together for Elizabeth and Anna.
It's an absolute pleasure to be here in person. It's fantastic, isn't it? And it's also a great honour to be speaking as part of the Mary Beath Memorial Lecture. I don't know how many of you know the work of Mary Beath. She sadly died in 2012, but I had the pleasure of meeting her just once, but I know her work quite well. Mary was a fantastic investigative journalist, and she in fact won several awards for her work there. She was also a very rigorous researcher who spread her net quite widely before sort of homing in on aspects of Highlands and Islands culture, particularly traditional medicines and sort of folk medicines of the Highlands and Islands and the Celtic tradition. And having met Mary once, I was saying to Elizabeth just yesterday, I could really imagine that she would have enjoyed the conversations that we've been having. And we would certainly have enjoyed, Elizabeth never met Mary, but we would have enjoyed sitting with Mary with a wee dram in front of the fire, and I think she would have been a very robust and engaged conversation partner. I think she would have enjoyed this. Our exploration of soil and plants and the human microbiome.
So one of the organisers tonight said, do you realise you have a typo in your slide? And initially it was a complete mistake, but we did realise, and we decided to leave it, because tonight we're putting the I back within the microbiome. So we're deciding to leave it there.
We did have quite a laugh.
We did have a laugh.
I hope you enjoy the humour in that too.
So this is also the Year of Stories in Scotland, so we're going to begin with Once Upon a Time. So once upon a time it was said that the only good microbe is a dead microbe. And microbes until recently in the mainstream have had a pretty bad reputation. We've been told to get rid of them. This slide is actually from 1910, and it's from the Wellcome collection, and it's people using anios disinfectant to destroy microbes representing infectious diseases. It's interesting how they depict the microbes as wee monsters, and would make you believe that no one had ever seen a microbe before, which is completely untrue. So we're going to go on with Anna, who's going to continue our story, a little bit about the history of our relationship with microbes.
A very potted history, and very much Europe-centred as well. So I'm going to talk about the term animalcules, little animals. And this was a term given to the discovery, so these little microbes that were first seen by Anthony van Leeuwenhoek from Delft in the Netherlands in the late 17th century. And he made an early version of a microscope. He wasn't the first to do that, but he was the first actually to observe these microorganisms, protozoa particularly, bacteria, and further on he was able to also observe spermatozoa. He was also the first to give us a really accurate description of red blood cells that any scientist nowadays would fully accord with. So he found these animalcules in lots of different sources of water. He was a prolific experimenter, and also tested this equipment on samples from the saliva from humans and animals, from the mouth and different parts of the body. And amazingly, he was also able to calculate their sizes and their numbers, and much, much more. He effectively laid the foundations for several disciplines of microbiology in Europe. And he was delighted to observe these little animals, as he called them, he became quite a celebrity. He had kings and queens visiting, and all the other famous people of the day came to his laboratory to view these animalcules, and they were absolutely amazed. It was almost like a theatre that they were coming to.
So what went wrong? We went from such wonder into...
Yeah, from wonder to horror, really. So basically, if you imagine people at that time, there were great waves of movement into urban areas and away from rural areas. There were several waves of this going on throughout Europe, not only in Britain. And with the urban development, you got much more crowding in very unsanitary conditions, and you had massive outbreaks of deadly diseases. Things like plague, but also smallpox and polio, diphtheria, some of the diseases that we have largely combated, we've eradicated pretty much with vaccinations. Tetanus and typhoid and cholera due to contaminated water particularly. At that time people thought, or up until that point, people thought, it was a very old theory called miasmas, and people thought that germs originated spontaneously, or that diseases originated spontaneously from bad air. The name for malaria comes from malaria, it's a bad air. Miasma was another term for it. And then gradually, as we go into the Renaissance, we've got several developments that are really crucial. They're just listed here, just to run through them very quickly. In Italy, there was a real flourishing of scientific research among physicians, amazing polymaths. Fracastoro discovered contagion, particularly in his studies of syphilis in the 1530s. Other scientists in Italy were discovering, or exploring rinderpest, which was a big problem among livestock, and in Slovenia as well. This theory was expanded. So you have all these developments going on. Edward Jenner, you'll all be familiar with, studying smallpox and finding that he could effectively eradicate it or treat it using using samples from cowpox. But he didn't know how it worked. That was still a complete mystery. Then further developments in public health, in hospital care with antiseptic procedures, with Semmelweis in Hungary, with obstetrics, Lister in advising the use of carbolic acid, carbolic soap, you may be still familiar with, before surgery, before practicing surgery. And then John Snow in London, who was really a pioneer, who managed to avert a cholera epidemic in London by identifying contaminated water sources. And then the famous Louis Pasteur in France, who managed to prove that there was a growing body of evidence for germ theory, that he was able to find proof. He then went on to develop vaccines for rabies and anthrax and won a Nobel Prize for his work on tuberculosis. Florence Nightingale, again you'll be all familiar with her, she was one of the first to apply these principles of fresh air, hand washing and improving sanitation, just sanitary practices in the hospitals. Then in Germany we have Robert Koch, and there's still a famous Robert Koch Institute in Germany carrying on this work, who managed to identify the anthrax organism. And he, sorry, I've got the details wrong here. He was the one that won the Nobel Prize for tuberculosis work. And he also was very crucial in that he invented the method that we now know of as bacterial culture, so culturing organisms in a petri dish, named after his assistant, Julius Petri.
So we've got back to the point where the only microbe is a dead microbe. But with the recent technological developments in scientific research, we've realised we need microbes. Those in our food and those in our gut, but we're actually more microbe than human. So how can that be? Anna's going to tell us a bit more.
No. I've got a little bit more on this. So one of the things that held things up in terms of discovering the good microbes was that it tended to be the pathogens, the bad ones, that grow in cultures. Now, some of these new techniques have enabled the detection of many, many more microbes, and this is just something that has thrown the whole thing wide open. One of the techniques is using ribosomal RNA and genome sequencing. PCR testing, we're all strangely now familiar with, not just in the world of science. And so this has gradually given us a better understanding of the microbes. And I've put these three terms, so their ubiquity, they are everywhere on the planet in every possible environment. They are important in all sorts of ways that we still don't fully understand. And they have enormous potential as well. And we're going to explore that a little bit. So this new understanding has led us right back to looking at the origins of life on Earth. In fact, all life on Earth came from microbes. So we're talking about over 3.5 billion years ago. The Earth was still a gaseous mass, but gradually settling down. And it's only been very recently, 2015, 2016, research in the Atlantic off the coast of Greenland has unearthed evidence of ancient bacterial strains, fossils, on the one hand, but also they've been able to extract some very ancient bacterial DNA from this structure called Loki's Castle, found in a hydrothermal vent deep under the ocean. About 2.5 billion years ago, cyanobacteria, blue-green bacteria in the ocean, started producing oxygen. And this started to change everything. Into this picture comes a fantastic researcher called Lynn Margulis in the 1960s, who put forward this off-the-wall idea, it was considered off-the-wall at the time, that bacteria were not only hostile, but they could actually work together, they could live together in what we now call symbiosis. And symbiogenesis was the process that's come to be known as the Great Engulfing, which basically means you've got one microbe, one organism, engulfing an aerobic organism, and they fuse basically, and they...
Is that like our mitochondria?
And
It's
they
like a horizontal gene transfer.
That's the one. I knew I was missing something. There we go. Yes. So basically, that is the start of the origin of all multicelled organisms, eukaryotic organisms. So, some of those then went on to fuse with other organisms that formed the chloroplasts, which give plants the possibility of photosynthesizing. But that's another whole story. So basically, in short, bacteria rule the world.
And we live with microbes. And we need to find now a different way of living with them. We need to look after them, and we need to provide them with a cosy home. And this is what we're going to go on now to talk about.
Here we go. So, where are we now? Microbes and us. So, as I said, many more organisms than we ever thought in every environment on the planet. Microorganisms don't act alone, they form communities and they collaborate. And this has implications for how we study them now and in the future. And not all microbes are pathogens. So fewer than 100 species of microbes are actually harmful to humans. Out of about 1.5 million that we've discovered and identified so far, and there are many more, as I said, still to be uncovered. This gave rise to the hygiene hypothesis. So looking at urban development, post-war development in urban centres in Europe particularly, you suddenly had a massive explosion of atopic diseases, things like hay fever, asthma and eczema. So a guy called David Strachan in the late 80s put forward this hygiene hypothesis that the problem was to do with lack of contact with farm living and animals, smaller family sizes and living in modern buildings, away from nature basically. But this hypothesis had its limits, and in 2003 Graham Rook came up with a hypothesis that embraced the hygiene principle but called it the old friends theory. He is an evolutionary biologist, still very active, but he took an evolutionary perspective and worked on the principle that all mammals, original mammals, 210 million years ago in the time of the dinosaurs, were burrowing creatures. They lived, breathed, consumed soil and dust. So they have evolved, all mammals have evolved, a set of bacterial symbionts, if you like. So they are part of our DNA, they are part of our theories are still evolving, so watch this space. But basically, we can sum it up as, there is no us without microbes. We are more microbe than human, as Elizabeth just said.
So we have to respect the microbiome.
Yes.
Because we're more than 90%. And as Anna said today, that these guys are kind of like your key workers. They're going to be taking out your rubbish. They're going to be tending your ailments.
Recycling.
They're going to be recycling.
Yeah. Caring, so the care workers,
yeah.
The refuse collectors,
yeah.
all of that.
So
high time we looked after them.
It is, but only mainstream medicine has only been looking at gut microbes in about the last 10 years. We've been looking at the soil and plants for a lot longer, but it hasn't impacted on the way that we're growing our food. Until recently, we're going to move on to that. But the microbes we're talking about, they include bacteria, archaea, not IKEA, but archaea, viruses, fungi, protists, and parasites. So we're going to look at the microbiome, but we're just going to define, first of all, what is a biome?
What is a biome, indeed? When I was putting together this slide, I came across all sorts of hits that cropped up on Google with biome in it, and it was nothing to do with this. It was all sorts of products being marketed with this new buzzword, biome. So I'm not going to read out this definition, you can read it for yourself. So basically, a biome, if you think of the rainforest, the desert, savanna, these are the classic biomes, these biogeographical regions with their populations of flora and fauna that are adapted to that environment, and they have several habitats and niches within those. The microbiome
is a community of microbes. So, for example, in the gut, in the soil, and in the air, they don't exist alone. They're all interconnected in a kind of super-organism. So we're going to look now at the communities. They all need the right environment to survive and thrive. And this next person we're going to look at is Tim Spector. And it's through his research that light has been shed on the importance of the gut communities and how we treat them for health.
I'm just going to rewind a tiny bit, Elizabeth, because the human microbiome is regarded as having four main components. So there's the skin microbiome, there's the airway microbiome, the genitourinary microbiome, and the gut microbiome, which is by far the biggest, and so far it's been the best, the most studied as well. So that's why we're kind of homing in on this one. It's also become very, very well publicised through the work of Tim Spector, his books, and also you may be familiar with the Zoe project that has been doing a lot of studies relating to COVID and how we can understand how our microbiomes are responding in helpful or unhelpful ways to novel pathogens like SARS-CoV-2. The human gut microbiome, as Tim Spector says, should be understood as an organ in itself. It's actually telling us a lot more than DNA ever could about health, particularly things like obesity, propensity to diabetes, to autoimmune disorders, all sorts of things. Microbes are using what we consume as food and drink to make substances that are absolutely essential for our proper functioning. Things like vitamins and minerals, and many of our signalling molecules, which are really important for our mood and for our endocrine system function. And those are areas we'll maybe come back to a little bit. So these microbes interact with food, and what they've also discovered is that every individual is very different. So individuals respond very differently to the foods they eat, depending on the population of microbes that they are host to. And the research is continuing all the time. So this science, the work of Tim Spector in this field has only been going for about 15 years, so it's very, very new.
What we know is that healthy people have a rich gut microbiology. But unfortunately, a Western diet of processed food isn't supporting a healthy, rich gut microbiome. So you see on this slide, it's just a little cartoon. My lord, the humans' reliance on food delivery apps has weakened our defence sources. They need fibre. So this food that we're eating can feed or damage our microbes with consequences. So you need to look after your gut microbes, and they will look after you. So let's look a little bit more into our gut.
So I'm just going to run through very briefly the key findings from the British Gut Project, which was set up by Tim Spector in liaison with the American Gut Project and another researcher called Rob Knight. So it's now so the British Gut Project has been subsumed into the Zoe project. Please do look it up, there's lots and lots more interesting information on there. But these are the key findings. So diversity is vital. So microbial diversity is influenced absolutely by the number and range of the foods that we consume, and especially the plant species that we consume. They've said that the best, the most diverse microbiome will occur if you eat at least 30 plants per week, at least. And those should be the usual suspects: whole grains, pulses, fruits, nuts, seeds, herbs and spices. But we can also include polyphenols, much to our delight.
Polyphenols, red wine, dark chocolate, and coffee.
Yes. See you in the art club later.
See you.
So the other thing is to ensure fibre intake. Now we've known about this for a long time, but to keep us regular. But we haven't really understood just what an impact this has in terms of how it interacts with the gut microbes. So it feeds them. It maintains healthy diversity. It feeds the good microbes, which helps them crowd out the bad. It helps our immune system, because the fibre, where it feeds some of these beneficial bacteria, is to create short-chain fatty acids, which act as anti-inflammatories. So they are crucial in the way that our immune system responds to pathogens and to attack from other things as well. So this emulsifiers and artificial sweeteners in manufacture. So we've been hearing a lot recently about ultra-processed foods, so that's really what they're talking about here. So they make the gut bacteria produce dangerous substances which can actually adversely affect our health. And the effects of these are much worse than previously realised. And basically everything else is okay. You can have meat, you can have dairy, you can have a bit of this, a bit of that, whatever takes your fancy. Have your cake and eat it. And it's all okay, but in moderation, which is perfectly good sense. But everyone's gut microbiome is completely unique, even identical twins. Their DNA may be 99.9% identical, but their gut microbes will be hugely different. And the future is that there'll be no diet books that say, eat this, do that, follow this plan. It will be individualised. It will be absolutely personalised according to your microbiome. But this is only one area, and it's very important, but it doesn't tell us very much. It's just very generic advice, if you like, very simple, about how our food is being produced, what foods we're actually eating, where they come from.
This is my garden produce. Before I came up to Orkney, what I'd gone out and picked for my lunch, and we'll then see this in a bowl later on. So what makes food healthy and nutritious? Soil. We need healthy living soil. Because now we're discovering hundreds of trillions of microbes that are part of healthy soil. One handful of healthy soil, 9 billion bacteria, and within that handful is 2,000 miles of fungal hyphae. There's going to be 3.5 million protozoa and 350,000 beneficial nematodes. That is a lot in a handful of healthy soil.
It's quite hard to imagine, isn't it?
Yeah. I mean, it's the hidden half of nature. It used to be a black box, and now they're saying it's just a grey box. But scientists still can't, what's the word? Emulate soil in a laboratory. There are so many connections we're not even aware of yet. So it's teeming with life, with bacteria, fungi, protozoa, nematodes, and many other micro and macroorganisms. And these are all working together, living, dying, eating and being eaten. And they live in cooperation with plants and their roots and supply many of the nutrients needed for them to thrive. And this mostly happens in a plant area. This is actually a plant area called the rhizosphere, which is 2 millimeters wide. This is just new technology that we can actually see what they look like in an electron micrograph. So this is happening in a plant area called the rhizosphere, which is kind of up to about 2 millimeters wide around the plant root. This slide is taken from work by Professor David Montgomery, who is actually a geologist, but with his wife Anne Bilks, who is a biologist. They've created their own garden, and for 10 years have been gardening in a microbially friendly way, and this is all part of their research. He calls this the biological bazaar, and I said, why does he call it that? Because there's so much trade going on. This biological bazaar of trade between the plant roots and all the microbiome and microbiota around it. Is this a new discovery? Surprisingly, it's not. It was actually in 1904 that a man called Lawrence Hiltner introduced the term rhizosphere. He said, I quote, that beneficial bacteria are not only attracted by the root exudates. And
what are those again?
What are they again? So exudates, so the plant is photosynthesizing, making sugars and carbohydrates. The plant is giving up to 40% of what it's produced out of its roots. We used to think they just leaked out randomly, but new research is saying the plant is actually deciding when, where, who, which microbe these are actually going to. Hiltner carries on and says, but they actually adjust where the specific root exudates are going. And he hypothesized that the resistance of plants towards pathogens is dependent on the composition of the rhizosphere microflora. Back then, the term microbiome hadn't been introduced. He used the word microflora. He even said the quality of plant products may be dependent on the composition of the root microflora.
Way ahead of his time, wasn't it?
Way ahead of his time. And actually through this research, we found a lot of people who were way ahead of their time, but who have been ignored by the mainstream, but that is a completely different talk.
We'll come back next year.
We'll come back next year if you want to know more. This is the slide of the rhizosphere in action. So plants have a complex association with the soil microbiomes, and these are beneficial for plant growth, nutrition, stress tolerance, and getting rid of plant pathogens. Some even go inside plant cells, like the bacteria which convert or fix nitrogen from the air. So if you've seen on legumes when they've got the nodules, that's nitrogen-fixing bacteria that are working with the plant. And also, you know, that the hyphae of the maybe you all have heard of the mycorrhizal fungi. The plant actually invites the hyphae into the cells, and it's a bit like, I imagine it, if I'm going to the petrol pump and I'm putting the fuel in my tank, that's what the hyphae are doing. And the hyphae of the mycorrhizal fungi, it's vastly extending the plant network. And so the hyphae and the mycorrhizal fungi are getting the moisture and the nutrients from a huge area, and in return, the plant is giving it the exudates.
It's absolutely fascinating. It also kind of mirrors the discoveries that Suzanne Simard has been making in Canada in forests with the wood wide web, she calls it, the network of mycorrhizal fungi.
And these are very important. Sorry. They're really important because these are 90% of the land-based plants depend on these for bringing up minerals like phosphorus. So a well-functioning, and this is really important, a well-functioning microbial rhizosphere is key to imbuing our diet because we're imbuing the plants with minerals, vitamins, and other nutrients we need for lifelong health. But first of all, this has to be in place for our plants to be healthy. So this next slide is of kale in my garden that I've grown this year. But this quote is taken from the work of David Montgomery and his wife, Anne Bilk. Again, they've had this 10-year process of developing their garden. And they decided with friendly microbial gardening practices, they sent us away to be tested for its nutrient value. And it came back that the kale grown with good microbial practices had 2 times as much calcium and zinc as the USDA reference value and 4 times the amount of folic acid. They're trace elements, micronutrients, which is really, really important. But they're very neutrally good for us. And you've got a little bit more to say on this.
A little bit more. And this research comes from this book, doesn't it? So the David Montgomery and his wife Anne Bilk. And yes, and they started, I remember looking at a video of them, they showed the kind of soil that they started with in their garden. It was just dust, it was nothing. It had been under concrete, completely dead. So they really achieved a huge amount. So basically, just to summarise then what Elizabeth's been saying, the nutritional quality of our food depends on the biodiversity of the soil microbial life. It depends on this directly through the soil and the plant's microbiota, and also through the minerals which are solubilized by microbes and by fungi, particularly mycorrhizal fungi. It also depends, the quality depends also on these microbes indirectly. For example, if we eat meat or dairy products that are from animals that have been fed on plants grown in richly biodiverse soils. There's much more going on too, but we're going to come back to this. But we're just going to take a little detour to look at how we're actually growing our food right now, so the current agricultural practices and the effects that those are having on the quality of our food.
This quote by Dr. James F. White this is a research university in America. If we did away with our modern food industry, we'd be better off. So I'm going to explain why that is. This is a picture I took this summer of Suffolk. This field, you can just see a couple of onions, red onions in the foreground. The onion harvest has just been taken. This is a conventionally farmed field. The soil here was like sand, it ran through my fingers, there are no worms. You can see the edge of the land there, which has been left bare, it's for a green corridor. You can see nature, if we leave nature, it never leaves the land bare. You can also see in the land there the ruts of tractors. So big machinery, as you know when you've seen tractors, and in fact, the largest John Deere fully loaded combine harvester is as heavy as 6 elephants, apparently. It gives you an idea. And what's that doing? That is pressing and compacting the land. What we know with microbes and the plant roots is they need air spaces, they need pores to grow in. So, and in a conventional system, they'll be using herbicides, insecticides, pesticides, fungicides,
molluscicides.
So I'm going to go on to the next slide, which is a study done from 2018. It was an academic paper which compared regenerative farms and conventional cornfields of 20 farms in North and South, the Northern Plains in America. So you've got, I'm going to go further into this because tonight I would really like you to go away with 8 principles of if you are going to grow your own food, of how you can do that. So a regenerative farm has got no-till, which is they don't plough, it's no dig. They're not using any kind of chemicals, no insecticides or pesticides, herbicides, fungicides. They're using cover crops. Here there's predominantly clover. So that the land, in America they say the land is armoured. So the land is always covered with a diversity of plants. And they use a rotational system. It's never the same crop all the time. They're more basic polyculture rather than monoculture. The conventional system, they plough at least once a year. They're using insecticides, they're using chemicals. They are leaving bare soil. They take one harvest out, leave the soil before they put another harvest in. And the results of that study were quite phenomenal. That the conventional practices had 10 times as many pests as the regenerative farmers. And although they had a lower grain production, they had 78% higher profits. Because they're not using the inputs, they're not using the heavy machinery, they're not using the fossil fuels, they're not paying out for fertilizers. And all the prices of fertilizers and insecticides have rocketed. And the profit was positively correlated with the particulate organic matter in the soil, not the yield. Organic matter is absolutely key. And I just want to read you the conclusions of the researchers of the paper. The farmers themselves have devised an ecologically based production system comprised of multiple practices. Regenerative farms fundamentally challenge the current food production paradigm. By promoting soil biology and organic matter and biodiversity on their farms, regenerative farmers required fewer costly inputs like insecticides and fertilizers and manage their pest populations more effectively. Soil organic matter was a more important driver of proximate farm profitability than yields. This ecologically based production system comprised of multiple practices, and these are principles that we can use in our own gardens because they are providing the right conditions for the plant microbiome around the rhizosphere. So, we've seen these ways of looking after our soil and growing our food, and we're going to go into that in more detail, but first of all we're going to look at how this affects our human health.
And it does very, very much. So basically what we're looking at here, I said we'd come back to this after Tim Spector. The idea of what is a healthy diet, even as proposed by Tim Spector and his team who are doing all this amazing work, we need to expand that a little bit, or hugely I think actually, being very understated there. So we need to expand what constitutes a healthy diet to include, as far as possible, food grown in healthy, biodiverse soil with a rich microbiota. And this is just a diagram of the difference between the two. So on the left, you've got conventional agriculture with herbicides, pesticides, fertilizers, etc. You've got a fragile plant microbiome and a poor soil microbiome. All these things go together. Then you have conventional food, which is produced in these conditions, a diet that's then rich in meat, low in fresh fruit, salads, raw vegetables, may have pesticide residues, and it results in a poor microbiome. Okay? So the microbiota in the foods that are produced in this system result in a poor and very undiverse microbiome in the gut. Healthy food grown in healthy soils from a rich microbiome, you have a healthy plant microbiome, using sustainable agricultural techniques, and then you have healthy food which then contributes to a healthy, diverse gut microbiome and has positive, huge, wide-ranging benefits for overall health, particularly immune system health, but all sorts of other things, which we'll come back to. So this is the link back to that. So we need to expand what we think of as a healthy diet. This slide is from a project by an artist that I've worked with in Edinburgh called Natalie Taylor. She did a residency for a couple of years with Northlight Arts in Dunbar, and it was called Is Soil Alive? And she was collecting soil samples, bringing together art and science. And she was analysing these soil samples from several different locations around East Lothian, near Edinburgh, in a laboratory with an electron microscope, and taking images from these, and then using those to create artworks, and to create conversations, to start conversations with young people in schools, people in the local community, in community growing projects, about all of these. And her starting point was that we are losing soil all over the world at a phenomenal rate, not only losing soil but biodiversity, but soil per se. And if you've ever been to one of the big urban centres like Glasgow or Edinburgh recently, you'll see how much soil is being built upon and sealed. So we're losing that as well. And with it, that diversity.
And with it, our relationship with a living organism. This next slide is the food that you saw before from my garden. This is my lunch. Straight from my garden onto my plate within half an hour. It is the most amazing, empowering feeling. I'm hungry, I just go out and pick. And in my garden now, I've got over 30 varieties of vegetables, fruits, and between May and up till now, I'm really not buying any vegetables. So you're probably thinking, well, it's all right for her. She's got a big garden. She's been there for ages. It's really well established. I can't do that. I only moved into this property.
You see, folks.
I moved into this property last October. It was completely paved. This is the top picture in February. I decided I can't live like this. I can't live with concrete. I need plants. So I just set to with my spade, and I've taken out 4 tons of concrete of this top one. At one point I did ask the help of 2 very friendly builders who had an electronic lift because I was a bit tired after taking out 2 tons of concrete, and I got them to do the last 2. So you can see the transformation between February and July of this year. The most important thing I know as a gardener is organic matter. And I knew I've just got to get as much organic matter in here as possible. So I'm fortunate being a gardener that I work on a lot of big properties that has a lot of dead wood and woodfall, and the owners are very happy for me to take what I want. So I have some deadwood in there, which will give a certain type, the saprophytes. The saprophytes. I did look for roadkill, because that's a great source, but I couldn't find any. So I went to the green, your composting green waste when you put out your green bin. That's on an industrial composting process, and you can just buy that in. I paid, some people say it's really expensive, that was £20 for a cubic metre. So I ordered in 5 cubic metres. So 5 big bags, and I set to and put it in this garden. So, as I said before, there are principles, what you can do, and as you've seen with the studies, the correlation between organic matter and yield and profit. Organic matter is so important because organic matter is the food. It's the food of the soil food web. It's the food of the microorganisms. If we don't have organic matter, they've got nothing to live on because they're solubilizing. They're taking all the trace elements and minerals of all this organic matter and then they're producing it in a plant-available form. So the relationship we looked at before in the rhizosphere, this relationship, this biological bazaar, this trading going on, they're basically the plants saying, well, I need zinc, I need magnesium, I need copper, I need this, and they're going, well, I've got it, and you're going to give me some sugar in return. So this is, you may have heard of the soil food web. It's becoming a more popular concept. It's just basically the recycling of nutrients, but the beginning of it is the organic matter. On the right, when I dug everything up, I found 6 skinny worms in the soil. So you might think, how do I know if I've got a garden full of healthy microbes? Just look at your worms. If you've got big, fat, healthy worms, they're part of the soil food web, you know you're going to have a healthy soil food web. So I knew I didn't have one at all.
Do you remember there was an article where somebody had done an experiment with 2 different types of soil, a very non-biodiverse soil, so with very little organic matter, and one that was rich in organic matter, and buried 2 pairs of cotton underpants, one in the poor soil and one in the rich soil. And after a month, they...
5 weeks.
5 weeks.
Complete disintegration. If your cotton pants have pretty much gone in 5 weeks, you've got great microbes.
So they've been biodegraded very
Go
effectively.
home, bury your cotton pants. So up here, organic matter is really important. So this morning I did a composting workshop at Scale House, and they shared their gardener's diary with me, and I was going through it. There was no mention of composting, but there was mention of dung.
Well, this guy is saying, believe it on here. So it says, all this day I was dunging and digging in the west border. John Taylor drove 1 load of horse dung and 6 of cow. Chaim O'Hesker, Karen Carcandu, which is Gaelic for my afternoon turned a bit shitty.
He does a lot of dunging. And then there was no car road into the property. So, you couldn't bring anything by car, so everything was brought in by car. He did a lot of this. He did a lot of dunging. And that's his organic matter. That's what they used. So these 8 principles, I'm going to go through them quickly-ish. So this next slide. So I've talked about the first one, hopefully in enough depth to realise this is the green waste compost being delivered. This isn't actually my garden, this is a client's garden, but it was a fantastic picture. The second principle, this is actually my dog, Mushka, and this was when I was sieving the concrete out of my soil. But this is actually saying, don't disturb the soil. It's an image of soil. I very disturbed the soil because I wanted to start with no concrete and no foreign particulates in it. So don't disturb your soil. You can imagine the plants and the rhizosphere and the mycorrhizal fungi. There's tractors that are ploughing up and down. They're breaking them all up, so they can't communicate together or with the plants. And that means we're losing that communication and the nutrient, that biological bazaar. The third one is plants. You need a great biodiversity. Each plant has, like we have a different microbiome, each plant has a different microbiome. So in the monoculture, it's going to be pretty much the same microbes. But if you've got more of a jungle garden, and there's a book by Goulson, he really says do a jungle garden. So when I planted my garden, nature doesn't plant in rows. So when I did my garden, organic compost, organic matter on, and then I got all my seeds that I had and literally just threw them on. Which is what another gardener, a regenerative farmer in America called Gabe does. And he said, my lunch is that I walk out and it's the first thing that I kind of trip over. The next one is compost. Make your own compost. You know where your organic matters come from. You can pump compost. Your microbes are local to your garden. Locality is really important. This one down below, these are actually poppy seeds at times 40 magnification. Seeds have their own microbiome, so try and collect your own seeds. It was only last year that a research was done at Stockholm University, and I'll just read a conclusion from their research. Research showed on oak acorns contain a large diversity of microbes, and that these oak seedlings inherit their microbiome from these acorns. The microorganisms found on the seed are often valuable for the plant, promoting its growth and protecting it against certain diseases. Each plant species harbours a distinct microbial community. So if you're starting off on a garden and you don't have any seeds, stay away from F1 hybrids and try and buy good organic seeds. There's real seeds, and I think up here there's Scotia seeds.
And you can contact your local community gardening project.
Or organic farm, or together swap seeds.
Yeah.
But if you can start off with healthy organic seeds, they're going to have their own microbiome.
It's interesting, just I don't want to interrupt you in mid-flow there, but I've seen written about a parallel between the seeds having their own microbiome if they're produced from a healthy plant. Some writers have drawn a parallel between that and a natural birth, and how
Absolutely.
a baby born by the natural means through the vagina is coated in the mother's microbiota, and is a better start in life.
So you're giving the plants a better start. Yeah. The next one is keep your ground covered. This is a cover crop of Phacelia. And the last one is, this was in my supermarket, this is the garden shop. Every product has got glyphosate in it. So we know glyphosate apparently doesn't affect human cells, but what it does do, it kills microbes. And we know that from studies. So if you're using any glyphosate-based chemicals, you're going to be damaging your microbiome.
Absolutely. And many of the others, many of the other products that you might want to put down as pesticides, insecticides, etc., they will also potentially have very harmful consequences for the plant microbiome and the soil microbiome. We're not quite done there. We've got a little bit more to share with you. Because it's not just what's going on under the ground, in the root system, the rhizosphere that we talked about. Plant power is not just about the nutritional aspects of the food quality. There's also a lot going on above the ground. So the plant, as Elizabeth said, has its own microbiome in every part of its being. The phyllosphere is the above ground part in general, and then you have what's called the endosphere, and within that you have endophytic bacteria. And those are communicating with the soil bacteria and with the rhizosphere as well. They're also communicating and interacting with the environment around about the plant, and absorbing microbes from the air and all of that, giving out some too. I happen to really like this particular diagram. You may recognise this plant as a nice local Orkney one, Scottish one maybe. Your kids might know it as soldiers, ribwort planting, but I just thought it was quite fun that we hit upon that one. So, plants are not just about food, and the plant microbiome is not just about food quality. Plant microbes make medicines as well. So, ethnopharmacology, and a discipline which is going probably since about the mid-1980s with people like Geoffrey Cordell and David Newman. They have been developing this branch called natural products research, so bioprospecting for plants that can be used as medicines. And we have many in our pharmacopoeia that are derived directly and indirectly from plants, and that bioprospecting by pharmaceutical companies is going on all the time, particularly in areas of the world which have high biodiversity, like rainforests. So plants produce certain compounds for their growth and development, and those are called primary compounds, or primary metabolites. They then go on to produce secondary metabolites, which are those that the plant may need to attract pollinators, to develop its colour, its scent, its interaction with the environment, its defence compounds, if it's feeling a threat, or if it's attacked by a pathogen. It's changing its chemistry all the time through its growth cycle, in intimate interaction with its environment and with its internal and external environment. And these secondary metabolites are what humans have harnessed for millennia as medicines, in folk medicine. We nod to Mary Beath here as well. And not just by humans. So I put this little picture of a capuchin monkey here. There was a wonderful research project run by scientists with the Edinburgh Zoo. When they put the food into the monkeys' enclosures, they also gave them onions and limes cut in half. And the monkeys didn't eat these. Can you guess what they did with them? They were rubbing them into their fur and each other. It's called anointing. And they did it as a social thing, but they were also rubbing it into themselves. And what they were doing was harnessing the antimicrobial and antiparasitic properties of these plants. So we have been doing this, mammals have been doing this for a long time. We recognise the compounds. These monkeys didn't have access to onions and limes in their native environment, but they know that they sense that these have properties that can be used for a particular purpose, quite specifically. So, now we're just beginning to understand that these secondary metabolites are not only produced by the plant, but they're being produced by the endophytic bacteria that live within the plant. So they're produced by the plant's microbiome. I've got many, many examples that have already come up with this. So Taxol from the yew, vinca alkaloids. So we thought until now that these were being produced by the plant, but we now know that the microbes are intimately involved with this. So we're back to the microbes. So very, very briefly, again, like what we did with the human gut, we're going to zoom in just a little bit. Some of these microbes are now being researched for their potential benefit to human mental health and well-being. This guy, Christopher Lowry, holding up a bunch of spinach, saying there are 800 plus microorganisms in this spinach growing in good soil.
We had spinach for tea.
Yes, we did. Yay. And this microbiome, so the microbes in the plant and the microbes in our airways and in our gut, they can affect our mental health. They act in all sorts of ways through this gut-brain axis, which is being researched. Several microbes have been identified as having this effect. And there's lots more research being done. So we're just wondering, again, back to soil. These soil microbes, could the solution to mental health problems be right under our feet all along? Again, very, very briefly, soils have been actually used very directly as well. So not just through the plants or through the foods. There's a whole branch of nutritional science, nutritional mental health science as well. But soils used directly all around the world. It's absolutely fascinating. I went down a rabbit hole with this, but I'm just going to show you very quickly.
But basically, we've kind of known a long time ago that we needed soil. And you've got some examples here of where practices where people have used soil without knowing why, and a bit like Mary Beath, people have gone back now and analysed that soil, and what they have in common is they all have the same bacteria in them.
Yeah. They have. But these particular ones that I've listed here, that are very traditional folk medicines that have been researched, and there are many, many more, they have all yielded new strains of Streptomyces, which is one of the major families of soil bacteria, a multitude of strains of these. And it's really interesting that they have this in common, and some of the uses of the soils that the soils were put to in traditional medicine all around the world. Many antimicrobials have been developed out of these Streptomyces already. But our relationship with the soil really runs deep, as I think you can see from what we've been talking about. So in addition to the science, we're also looking at some of our instincts. So our relationship with the soil goes back a long way, as our relationship with plants does, and recognising some of these compounds that can be beneficial. Geosmin, you may have heard of, is a compound that contributes to the earthy smell of rain, of soil after rain. You know, if you go out after rain and you get a lovely fresh scent, yeah? It's called petrichor, and geosmin is the compound that's at the root of that. It's also on beetroot. It gives it that slightly earthy taste as well that some people find quite unpleasant. But it's produced, I've said it's produced by dead microbes. It's actually produced by microbes in a dry period. Some of these microbes, Streptomyces for example, they die back, and in doing so, before they die, they produce spores. And those stay in the soil, and the minute you get some rain, that activates these spores, and you get this scent. Now the interesting thing is humans can sense this at 5 parts per trillion. I'm just going to leave you with the question, why?
Which is more powerful than a shark can smell blood in water.
1 million parts.
Yeah,
1
so
part
I was
per million.
amazed
So
at
this
that.
is 5 parts per trillion.
It's a
quite
super
something.
sense.
So I'm just going to leave you with a bit of a philosophical thought. The etymology of human. And like the name Adam in the biblical, in Aramaic, from the earth. Human means of the earth. Humus is earth that's rich in organic matter. And humility comes from the same root, where maybe humility in our context means recognising that we are soil.
And I've actually realised when I did my 8 principles, I only did 7, because the 8th was understanding humility and having the ability to relearn and reconnect with the soil and where we're living.
It's a long-term project, isn't it? So
Yeah.
learn, observe your garden.
Absolutely. So we just want to leave you. Thank you very much for listening. And these are just some book choices. I really, really recommend The Hidden Half of Nature. It's kind of like everything you could possibly want about microbes, and it also has the journey of Professor Montgomery and his wife, and Bill Cliff.
Yeah. Thank you very much,
Thank
everybody.
you.
I think we have time for a couple of questions, and George is just going to move around with the microphone. So if you do have a question, pop your hand up, and Georgia will present you with.
Thanks. You were saying about how it's good to keep the soil covered. So when whatever you've been growing in your garden, vegetable-wise, is finished for the year, what would you suggest planting over the winter?
So green manure. So it can be a clover. Clovers over the winter. You can even put field beans in, which will then grow over the winter, and you can harvest them in May. So find a green manure, and I'm being a little bit reticent because it will also depend on your conditions. Do you have clay soil, sandy soil? Where do you live? Do you live in Orkney? Do you live somewhere where it's going to frost more? So search for organic green manure seed. But you can always underplant. So I was reading this, something I've done this year is underplanting my beans and peas with bird's foot trefoil. I've got another area of red clover. I've got another area of phacelia. But just keep your ground cover with green manures.
Living out in the country, farmers spread slurry.
Yeah.
Yeah. Is that a good thing?
Personally, I don't. Howie and I have often talked about slurry, but slurry is kept in slurry pits, which is an anaerobic process. All these microbes are aerobic. And
they need oxygen.
They need oxygen. Slurry is anaerobic, kept in big pits for a long time. It's very acidic. When it's sprayed on the fields, it can often burn. So personally, I wouldn't use slurry, and I wouldn't advocate its use. If we're looking again, first principles back to nature, a lot of these regenerative farming are using livestock in a form called mob grazing. They're trying to reproduce what would happen naturally, like with bison and big creatures moving across the landscape. So they use cows with electric fences, and they're moving the cows over the grass. Well, the cow's poo, it's just cow poo, fresh cow poo, which is then dung beetles come in, and the microbes come in. It's not being kept in vats for months in an anaerobic condition. So if I'm just trying to apply the principles of what is a good environment for the microbiome, my mind is, having not researched it, my mind is kind of, I don't think it is great.
It's also worth mentioning that it should be cow poo that is not from animals treated with antibiotics to fatten them up. We read the quote from the gardener Hugh Ross from the Scale Garden getting on his dung in. Another issue now is a lot of animals are wormed with very strong wormers, which are killing worms, parasites, bacteria, nematodes. So if you think after this, I'll go down to my local
I don't know, horse yard and I'll get a lot of horse poo, you need to ask them, what are you worming your animals with? And some people say leave it for 6 months a year, but then you'd have to read which wormer it is. Does it biodegrade? Is it going to affect my? So it's a new and vast area, the research that we're finding, and it can be quite challenging, but we can only do our best.
Yeah, indeed.
Hi, I know that it is more aimed at people who are actually going to grow vegetables. But what's the situation if you buy organic vegetables?
Basically, that's a great thing to do if you can, because the organic principles will basically be very, very similar to what Elizabeth was talking about, the regenerative system. So organic cultivation is based on soil fertility, enhancing soil fertility, working with natural predators, understanding the soil, understanding the plant and what it needs.
And not using glyphosate-based products.
Yeah, yeah. In the organic systems, there are permitted pesticides and fertilizers, because if you're farming organically, commercially, then you want to get your yield in. But if you're gardening, just in your own patch, then you don't need to do anything at all. You will not need anything, because if you've got good soil, the soil will look after the plants, and the plants will look after the soil and look after each other.
Yeah, and I was particularly thinking if I buy certified organic products, can I be confident that they would be okay from a microbiome point of view?
A lot, yeah. I mean, pretty much, yeah, absolutely. Yeah. I mean, they'll be quite different from conventionally farmed products.
Yeah. Mm-hmm.
And you may not. It's really interesting because they've done lots of tests where they've got groups of people to do taste tests to see if they can tell the difference between something that's organically grown and something that's conventionally grown. And very often people can't. But it's not just about taste. So it may depend on the particular plant's species, the variety that you're growing. But it's actually in what's going on underneath. It's that hidden half that we're talking about. It's what's under the surface that's going on that's going to be really beneficial. So it may not be evident in the taste, but yes, you'll get the benefit of very rich biodiversity there.
Well, any more questions, I'm sure you can catch them afterwards. Let's put our hands together once again for the microbiome. Thank
you very much, Finn, and thanks to all the technical team up there as well.